Bismillah ar-Rahman Rahim. In the name of Allah, the most gracious, the most merciful. We start another video on paper 4.3 of June 2023, and this will be the first video which might have 3 or 4 questions. As you all know, this paper is for 2 hours, but it's a very long paper and you need to concentrate very well, and you need to first do the questions that you know. So the questions that you know well, first do those. Then come back and do the questions which are sort of half clear and half not clear to you. Because you have to realize is anything above 62 out of 100 is an A grade. Because of course the grade boundary changes, but if you, even if you lose out, if you don't do 10 marks of questions and you get 62 out of 90, you only attempt 90 marks of questions. I don't mean out of 90, I mean out of 100, but you only attempt 90 marks of questions and you get 62, well, you've got an A. But if you spend 20 minutes on the question you don't know, and you waste your time on that and you get a 0 out of 10 in that question, so what is the use of it? So this is called exam skills. This is called intelligent exam skills. Please remember that. Now, as you can see, the question 1 is on respiration, which is usually a very easy question. The Krebs cycle was named after the biochemist Sir Hans Krebs, who worked out the sequence in 1937. Figure 1.1 is an outline of the Krebs cycle. Identify substances P, U, P, Q, R, and S, and T, and U. So what is P? Let's look at what is P. Yeah, P is the 6 carbon, so that's citrate. And then what is Q? Q is NAD. What is R? R is, you could have said NADH, or you could have said reduced NAD. So reduced NAD would have got you the mark, or NADH would have got you the mark. And what is T? T is PQRS. What is S? Sorry. What is S? S is carbon dioxide. And T is FAD. So T is FAD. U is, what is U? Reduced FAD. Or you could have said FADH2. So if you get all these correct, well, you only get 3 marks. So, and please remember, in bio there is no half a mark. If you get 6 correct, 3 marks, 5 correct or 4 correct, 2 marks, and 3 or 2 correct, 1 mark. So there's no 0.5 in bio. Then coming to the C part of the question, describe and explain the features of ATP that make it suitable as the universal energy currency. Now this question has come so many times. Small, water soluble. It is water soluble, so it can move around the cell. What does this mean? Any cell, whether it's an animal cell, is 70% water. So ATP, being a soluble molecule, can go around the cell wherever it's needed. So water soluble can move around the cell because cytoplasm is 70% water. So if it is not water soluble, how would it move around? It's just like if a car doesn't have wheels, well, how is it going to move? The second point is hydrolysis leads to energy release. When ATP, we say, is hydrolyzed to ADP, so a water molecule is added, and this water molecule is going to split into O and H and OH, and ADP is produced plus PI. So loss of phosphate, or we can say the hydrolysis, leads to energy release, and this is going to release in some energy being released. Something 30 something kilojoules of energy is released. Now, this is 30.5 kilojoules per mole. So the release of energy is immediate in small packets. It immediately releases the energy. It doesn't going to take time. Then it can be recycled. Recycled means what? ATP can be broken down to ADP, and then, of course, it can be again made. ADP and phosphate can be joined to make ATP, can be recycled, or you can say regenerated. These are very typical biological English words. Everybody must learn these. Then there's a high turnover. What does it mean, high turnover? There are a lot of ATP is made and a lot of ATP is broken down. All the day, your body is requiring ATP and then it's going to be remade. So, made, broken down, made. Just like you fill up your wallet with 1000 rupees in the morning, and by the evening you spend that money, and then again you take another 1000 and put it in your wallet. So, high turnover, and this is the link between the energy yielding and the energy requiring reactions. You see, there is the glucose is broken down to release energy, energy yielding. Some of you might not know this English. So link between energy yielding and energy requiring reactions. Active transport requires energy, but aerobic respiration releases energy, and that energy is stored as ATP. You go and work in the whole month, and then the first of every month you get a salary, and that salary goes to the bank. And then you take out the money from the bank and you spend it. And then the next month again, your salary goes into the bank. But if the bank does not give you the money, then how can you use that money? So please remember, all these points are very, very typical, and we've seen this in many, many questions. As far as I'm concerned, this was a very easy question. So once again, we just go through this. Water soluble can move around the cell. Loss of phosphate leads to energy release. Release of energy immediately, and the magnitude is 30.5 kilojoules per mole. Can be recycled, regenerated. Or you could have written the formula. ATP is broken down either way, the works in both. So you see I've done an arrow in this direction and I've done an arrow in this direction. So it's a reversible reaction, ATP plus inorganic phosphate, or that combines to form ATP. This is the link between energy yielding and energy requiring reactions. High turnover. And then the last thing is ATPase. Now please do not confuse this ATP synthase. What is ATPase? Let's look at this now. Enzymes are proteins that catalyze reactions. ATPases are enzymes that use the energy from the breakdown of ATP molecules. The energy from ATP is used for many important physiological processes, including active transport, movement, and synthesis. So, active transport, movement, and synthesis. Monomer to glucose to glycogen requires ATP. Glucose to amylose or amylopectin requires ATP. Glucose to glycogen requires ATP. So ATPases are enzymes that use the energy from the breakdown of ATP molecules. Enzymes are proteins that catalyze reactions. I hope everybody is clear on this ATPase. Please do not confuse this with ATP synthase. Now coming to question number 2, humans use antibiotics to treat bacterial infections. The increased use of antibiotics has led to an increase in the number of strains of bacteria that are resistant to antibiotics. The evolution of antibiotic resistance in bacteria resulted from natural selection. Outline how bacteria become resistant to antibiotics. Number one, mutation. Number two, it's directional selection. Antibiotics act as a selection pressure. Bacteria with mutation have selective advantage. So these bacteria survive. Bacteria divide by binary fission, or it is asexual reproduction, or you can say vertical transmission. So the transformation, the will of course it will going to transform, but there can also be horizontal transmission. You see, vertical transmission and horizontal transmission. Vertical means that this bacteria divides, so both of the bacteria contain. This is vertical. Horizontal means there's another species of bacteria and the plasmid with the resistant goes into this one. Previously, it was just going into this one, those which were formed by binary fission, but now it's going to another species of bacteria just by horizontal transmission. Then the allele frequency increases, so you get a population of bacteria which are resistant. Then this is fast due to short generation time. Bacteria divides every 20 minutes. So it's very fast due to short generation time. Increased chance of resistance if people do not finish full course of antibiotics or they overuse of antibiotics. So they keep on taking the antibiotics or they stop the antibiotics beforehand. All that results in, you see, those are the factors which are predisposed to then mutations. Bacteria, if you don't give them the full course of antibiotics, you give it for 2 days and you stop it, the bacteria that are left are going to mutate. And the reason why this happens is because Allah has given them this capability. The reason is that there are naturally occurring antibiotics everywhere in nature. So bacteria will mutate if they are exposed to a certain antibiotic. So when humans indiscriminately use antibiotics, you've got flu, take ciproxin. You've got a toothache, take augmentin. Well, you don't know what the toothache is because of, maybe it's the root canal which you need to get it done. But you just take an antibiotic. You've got some infection in your throat, it's a viral infection, and you say, okay, let me start taking augmentin. You're all very fond of, because you can get them off the counter. You can just go to the pharmacy and you can just go and buy it. I was going to say you go into the grocery and buy it. Sorry, you don't go to the grocery to buy medicines, you go to the pharmacy. So this is how you were going to answer this question. Now this is a very long answer. It's only 7 marks, but it's a very direct question, and I'm sure everybody could have got the full marks if they knew all the points. So when you look at the mark scheme of this question number 2, you have something like 11 points, and you could have given me any 7 of those. What are those? Random mutation number 1. This is directional selection. Antibiotics act as a selection pressure. Bacteria with mutation have selective advantage. So these bacteria survive, or you can say reproduce, survive and reproduce, or survive and reproduce. Now bacteria divide by binary fission. Now this can result in vertical transmission, which I have explained it to you. Bacteria divide into this vertical transmission, or it can be horizontal transmission. Horizontal transmission means that the plasmid will transfer from this bacteria, it will go into another species of bacteria. Then allele frequency increases, or you could have said gives rise to a population of resistant bacteria, because if they're all resistant, then all the bachas are resistant. So more bacteria, more bacteria, more bacteria, all are resistant. And why? Because it's very fast. Every bacteria divides every 20 minutes, so due to a short generation time. So it is very fast. So if there are 10 bacteria which are resistant, in a few hours, in 24 hours, there'll be millions of those bacteria which are resistant. Increased chance of resistance if people do not finish full course of antibiotics or they overuse of antibiotics. Some antibiotics may act as mutagens. So antibiotic, you can say enzymes that break down the antibiotic. So those of course will all result in natural selection of resistant bacteria. Now coming to question number 3, gene expression in a cell is controlled. When a gene is expressed switched on, the gene is transcribed. Transcription means DNA to mRNA. When a gene is not expressed switched off, the gene is not transcribed, so no mRNA will be formed. Environmental changes can cause some genes to be switched on or switched off. An example of control of gene expression in prokaryotes is regulation in the lac operon. The lac operon is a length of DNA that is made of different parts. Figure 3.1 shows a simple diagram presented the lac-I regulatory gene and the lac operon. Outline the main features of the lac operon, and this is 3 marks. Main features of the lac operon are what? The operon has a promoter, it has an operator, and it has 3 structural genes. LAC-Z beta-galactosidase gene, LAC-A beta-galactoside permease gene, and LAC-Y, which is trans-acetylase gene. I know it looks very weird, but well, you've got to just know these names. You better memorize it, write on a piece of paper and keep on paste it just in front of you where you sit and study. And if you look at it every day, well, of course you'll remember it. You can remember so many other things, but you don't want to remember anything of your course. Well, that's up to you then, if you want to be the Changad of the century. So it has a promoter, it has an operator. You just wrote that, promoter, operator. Well, you got a mark for that. There's 2 marks you got. And then 3 structural genes. Well, if you wrote 3 structural genes, or you named them, if you knew the names, if you didn't know the names, then you could just say that 3 structural genes. Or you named the 3 structural genes. I'll just give you the names so that you remember the names. It's important that you know the names. So promoter, operator, 3 structural genes, either you just wrote this or you gave me the names of all the 3. Beta-galactosidase, lactose permease, and trans-acetylase. Yeah, I know it's difficult to remember it, but do try and remember it. Then, of course, we come into the part 2 of the question, which says, explain the role of the lac-I gene in the regulation of the lac operon. The lac-I gene is what? What did they say in the question? You've got to understand that they said in the question there's something. It shows a simple diagram representing the lac-I regulatory gene and the lac operon. So lac-I is the gene which is always expressed. It controls the gene expression. It codes for the repressor protein. There's a repressor protein which gets attached here. Repressor binds to the operator, blocks the promoter, prevents gene expression. So RNA polymerase cannot bind to the promoter. Lactose binds to repressor. So repressor cannot bind to operator, or the promoter is unblocked and the gene expression may occur. So if you have to revise this, when you need to revise this, the lac-I gene is actually coding for the repressor protein. The lac-I gene codes for the repressor protein. This is the lac repressor protein which blocks this so that these are not transcribed. So the lac repressor protein. The lac repressor protein, please remember, attaches to the operator here, you see. Now this is the promoter, this is the operator, and then these are the 3 structural genes. So when you look at this question, explain the role of the lac-I gene in the regulation of the lac operon. The fact that the lac-I gene is always expressed. Why is it always expressed? I'm just going to revise this with you, don't worry. It's always expressed because it controls gene expression. You see, those genes which are the structural genes will not work. Controls gene expression. Why? Because it controls codes for the repressor protein. This gene, I gene, codes for the repressor protein. Codes for the repressor protein. Now, what does the repressor protein do? The repressor protein binds to the operator, or you can say blocks the promoter. So the repressor protein binds to the repressor protein binds to the operator. And when it binds to the operator, what does it do? It blocks the promoter. So RNA prevents RNA polymerase binding. Prevents RNA polymerase binding. So, but if you want it to be removed, then when you put the bacteria in lactose, lactose binds to the repressor. So the lactose binds to the repressor, and then what happens? Promoter unblocked. Promoter when unblocked, what is going to happen? Gene expression can occur. So the 3 structural genes will be made. Now let's look at this diagram. Now you know all this is the lac operon. What is the lac operon? It's made of the promoter for the structural gene, then the operator, then lac Z, which is beta galactosidase gene, then the lac Y, which is beta galactoside permease gene or lactose permease gene, and lac A is the trans-acetylase gene, beta galactoside. And these are classified as the structural genes, right? Then we have the promoter and we have the operator. Now here you can see this is RNA polymerase, poor thing. But when the lac repressor protein is binding to it, this is the lac repressor protein is binding to it, is binding to the operator. And that is being blocked. So lac repressor the RNA polymerase cannot now make mRNA here, mRNA here, and mRNA here. So the poor lac repressor protein is being blocked by the lac repressor protein is binding to the operator, but the RNA polymerase now cannot function. Now when this lac repressor protein is removed, now you see this has been removed. So the lac repressor protein here has been removed. When it has been removed, now the RNA polymerase can work, and mRNA for this, and mRNA for this, and mRNA for this can be made, and it will go to the ribosome, and those proteins or enzymes will be synthesized. Now the lac I is this promoter for, this is the regulatory gene. This is the regulatory gene which codes for this lac repressor protein. Regulatory gene must, just like you have a regulator in the fan which makes it fast or slow. So this is called the regulatory gene which regulates whether these 3 genes will be transcribed or not, or these proteins will be made or not. Because if the bacteria is in glucose, then why should we have these 3, why should the amino acid be wasted to make these 3 enzymes when there is no lactose? They're only made for lactose digestion and lactose assimilation and the use of lactose. Just like if you drink water, why should pepsin be released from your stomach? So please understand this. First you have to understand this. You need to go back to the theory video which I have done. I've done this in detail in that, and then you can of course come back to this question. Now the last part of the question is B part. The lac operon codes for inducible enzymes. Repressible operons code for repressible enzymes. Suggest and explain why it is an advantage to a prokaryote to have a repressible. Inducible means that which can be induced, which can, if lactose is present, then inducible. Repressible means to stop them. You see, what you have to understand, enzymes are made continuously all the time. Why? Because enzymes are proteins and needed for the cell. Now when there is end product inhibition, what does end product inhibition mean? That there are a series of reactions. There's one thing which converts to 2 and then something converts to 3. So there's an enzyme here and then there's an enzyme here. Now when this 3 product results in end product inhibition. Why? Because this end product is too much. We don't want too much of it to be made. So end product inhibition is done. Why? Is made until product concentration is too high. So we only need say 100 of 3. But now it is 200. So these 200, 100, the rest of the 100 will go and block this enzyme so that no more of 1 is converted to 2 and 2 is converted to 3 because we have enough of the 3. It's just like you need 1 bread in the house and you have about 10 slices of bread in that. But you don't need any more because you're only 3 people living in the house. So why would you go and get 10 breads from any bakery? So that is why there is this repressible operon. So how do we word this? Enzyme is made continuously, enzymes needed for the cell, but then at times we need to have the enzymes to shut down, and that is when we talk of end product inhibition. We finish the video here because we'll continue the rest of the questions in another video. So thank you very much, and please wait for the second video for the rest of the questions. Only question 1 and 2. So only 3 questions have been answered in this video. Question 1, question 2 and question 3. And the rest will continue in the next video.